Good afternoon, everyone. Today we're going to finish talking about functional neuroimaging, which is a relatively long unit, and then move on to talk about neuropsychology, which is a relatively short unit. Now, at the end of the last day, we talked about MEG, and that, in addition to ECOG and EEG and single unit recording, are relatively direct measures of neural activity. Right? In other words, the neural activity itself is being measured. Now, in contrast, PET and optical imaging and functional MRI are relatively indirect measures of neural activity in that they're not measures of the neural activity themselves, but measures of their metabolites or their approximate neighbors. Now, PET itself stands for positron emission tomography, and the positron refers to the opposite of an electron, right? So when you think about atoms that are molecules that are used by neurons when they're active, oxygen is a primary one, right? So cells love to use oxygen and glucose, and so the more active a group of neurons are, the more oxygen and glucose they'll consume, right? And so the use of PET scans requires the injection of a radioactive isotope such as of oxygen. Oxygen in its stable state, of course, is oxygen 16, right? So there are 16 electrons for one atom of oxygen. Now, instead, we're injecting an oxygen 15, which is a radioactive isotope of oxygen, and that when it becomes active, because it's consumed by the neural activity itself, it causes a collision between a positron and an electron, which then emits a pair of gamma rays that are emitted in opposite direction. So that is the emission part of the positron emission tomography. And the tomography part is the use of some circular detector array that is arranged around the biological matter being measured, that detects this emission of the gamma rays that are in opposite directions, right? So when they're gamma rays that are being detected simultaneously or almost simultaneously in opposite directions, that gets counted as a hit, right? So that, and then all other activity gets ignored. So this radioactive tracer, which then emits the pair of gamma rays, is used as a proxy for the increase of neural activity. And so just to show you what a PET scanner looks like, this is Bill Nye the Science Guy who was going into the PET scanner. This is Brad Wojtek, who is now, at the time was a researcher in the PET scanner and now is a professor at UC San Diego, but he was operating the scanner. And one of the good reasons for using a PET scanner is that PET is very useful for detecting radioactive isotopes and therefore metabolites, chemical metabolites of the brain. And so there are certain disorders and diseases that are associated with decreased metabolism, such as of oxygen and dopamine. So Alzheimer's is one of them. There's decreased metabolism of oxygen and glucose in Alzheimer's disease. And so when you're doing PET scans, it becomes quite clear that there's overall lower levels of metabolism in people with Alzheimer's disease. So at least before functional MRI came along, PET scans was the best way to visualize brain activity in a functional way. Now, some pros and cons of PET scans. Pros are that it has relatively good spatial resolution, at least compared to the methods that required a two-dimensional inference problem of a three-dimensional solution from a two-dimensional source, such as EEG and MEG. So it's a relatively good spatial resolution. It does require an injection of these radioactive isotopes that are produced by cyclotrons, and so that is it's a silent injection, which means that you can present sounds within the PET scanner, but it is slightly invasive, right? So you are injecting something into the artery, and because of that, it's a minimal. I would call it a minimally invasive procedure, and you do have to have to make sure that the body kind of flushes out these radioactive isotopes immediately after the use of the PET scan. And how does the body flush out these radioactive isotopes? Well, it gets peed out. It gets passed out of the body, and so if you're a research assistant in a PET scanner, you'll end up having to clean up radioactive pee. So that's a little bit less pleasant maybe for the researchers. And then another con is that it has a relatively poor spatial resolution, right? Because this metabolism takes much longer time than measuring directly the neuroactivity itself. So it does require some sensitive reagents. So of course the radioactive isotopes has its own half-life, right? So if you can't just let a can of oxygen 15 just kind of sit there, because it's going to become oxygen 16, right? Because that's the stable isotope. So you have to go to a cyclotron, run and get this oxygen 15, and then immediately inject it into the subject, and then immediately put them into the scanner, and then immediately clean up their pee afterwards. So so it's not the most convenient measure, but it is useful for mapping metabolism. So if you have in surgical situations, if you're trying to remove a part of the brain that's not active or that's particularly active, you can use that as a surgical mapping technique. Now, in contrast to PET scans, there's also optical imaging, which is the ability to use light, right? So optical information to image brain function, and this is becoming used more and more these days, especially for as a non-invasive, completely non-invasive device, and especially for small children. And we'll talk about why in a moment. There's a couple of measures that that we're using when we're looking at optical imaging. And they, in general, use the number of photons. That they use a combination of light source, which is emitting photons towards the head. Now that light source then is assumed to go through the scalp and the skull and the CSF towards the brain itself, which, depending on how much activity there is in the brain, it refracts the path of light, which then gets picked up by the optical sensor and the output side. So it always depends on this coupling of light sources and optical sensors. And there are a couple types of measures. One is absorption and another is intensity. So one example of a measuring device is a near-infrared spectroscopy, which is a relatively slow technique. It's a measure of light absorption, looking at light intensity at the detector compared with the source, right? So the idea is that if there's a lot of change in intensity at the optical sensor relative to the source, you can know that there's more activity going on. So it's a relatively high spatial resolution technique. It has medium temporal resolution, and that's because the light bending itself requires the change of hemoglobin levels, which are oxygenation levels, the amount of oxygen in the blood that observes the brain activity itself, right? So not the brain activity itself, but the amount of oxygenation level in the blood that has hemoglobin in it, and that amount of hemoglobin actually is what then changes the light and bends the light. And so because of that, the temporal resolution is not great. It's on the order of seconds, but it does give you relatively good spatial resolution on the order of millimeters to centimeters, but that's specific for and only for areas that are close to the surface of the scalp, right? Because if you're coupling these light sources and optical sensors on the surface of the scalp, you can't really hope to get information much deeper inside the brain. And so And for this reason, NIRS imaging is also optimal for children, partly because their heads are relatively soft. So a baby's skull, I don't know how many of you have looked at a baby's head, but they tend to have relatively soft skulls. They're not completely fused yet. And so because of that, it's easier for the light to shine through. And another pro is that when you put these pairs of emitters and detectors onto the baby's head, it's a relatively light setup. So compared to EEG, which tends to be a cap for the whole head, and certainly compared to MEG and functional MRI and PET scans, I mean the baby really has to sit very, very still for those. But for NIRS, the baby can move around a little bit and still get these pairs of emitters and detectors detecting their proxies for brain activity relatively well. And so it's relatively non-invasive, it's inexpensive, it's relatively portable, as you can see in this NIRS setup here. But the cons are that you can only get good resolution of surface structures on the cortex, right? So if you're interested in the hippocampus or the amygdala or the basal ganglia, which are relatively deep structures within the brain, you're out of luck, right? For NIRS imaging. And it is a measurement of blood oxygenation level, right? So it requires hemoglobin, which is a molecule that's inside the blood itself, and that gives you a proxy for oxygenation level. And so it does not measure neural activity itself. So in that sense, it's an indirect measure of brain activity. So just to give you an example of a good NIR study that was done on around 5-month-old infants. This was a study of infants before and after they've acquired object permanence. So I don't know if you've heard about object permanence from other classes, such as maybe developmental psychology, but that's the ability of a baby to know that an object should stay where it is, right? So if there's a pen in front of you, you expect that pen to still be there in a few seconds. So babies turn out not to know that before a certain age, around 5 months. But around 5 months, if you have an object, if you show the baby an object and then suddenly it goes away, the baby shows some measure of surprise, right? Such as by looking longer at where the object had once been. So this suggests that the knowledge of object permanence is something that emerges. Now, so and what this group has done is show some differences in hemoglobin levels, right, which is a proxy for oxygenation activity, which is a proxy for brain activity. What they saw was these changes before and after babies showed object permanence. By the way, if anyone knows who this fifth author is, Natalie Hirschlag, that's Natalie Portman, the actress. So she was an undergrad at Harvard at this point, and she was a co-author on an article. So you can be a Oscar-winning actress and be a neuroscientist as well. All right, moving on to functional MRI. When we're talking about MRI, which stands for magnetic resonance imaging, we can use MRI for structural and for functional imaging. Right? We've already talked about structural MRIs. We talked about T1 images, from which you can do voxel-based morphometry. We can also do cortical thickness measures. Deformation-based morphometry, we didn't talk about it so much this semester, but it's another way in which you can infer differences based on the T1 images. You can also do diffusion tensor imaging, which is another way to use an MRI machine, which looks at white matter instead of gray matter. Now, in contrast, we're going to talk about functional MRI today, which now function, of course, just means changes over time, right? And you can have task-related functional MRI. So the classic example is you have somebody lying in the scanner and doing some kind of task, and then also doing some kind of control task, right? So let's say if you're interested in what people's brains are doing while they're listening to music, you have them listen to music for a while, and then you have them not listen to music, right? And you do a comparison between listening and not listening. That's kind of the standard task functional MRI. Um, in contrast, you can also do resting state functional MRI, and this is becoming increasingly common just because of how we're thinking about the brain. It's kind of becoming more refined over the last few years, and this is a different kind of functional MRI in which the person is not doing anything at all other than staring at, keeping their eyes open and staring right in front of them. So when they're doing this, they're in a resting state, right? So that's why it's called resting state functional MRI or resting state functional connectivity MRI. And so what we're doing is essentially taking a video of their brain voxels, voxel by voxel over time, and then comparing how the different voxels fluctuate over time. Right? So we're looking at the connectivity patterns between the different voxels, between the different regions within the brain as it changes over time. And in both cases, functional MRI is measuring oxygen consumption in the brain. So this is one of those indirect measures of brain activity. It's what we would call a gossipy neighbor of the brain activity itself instead of measuring the brain activity itself, which is what EEG and MEG and ECOG are trying to do. And so because of that, it tends to have relatively low temporal resolution, right? Because it takes a few seconds for the oxygenation level to go up after the consumption of by neural activity itself. But in contrast, you can have pretty high spatial resolution, right? So you can you can be identifying changes in activity in millimeter level of resolution, spatial resolution in the brain, right? Because you can tune your voxel sizes to be as small as you like you can for them to be. You can have 2 by 2 millimeter cubed voxels, and so you can visualize activity at a much better spatial resolution compared to EEG, for example. So we'll talk about a lot of aspects of functional MRI studies. First, we'll talk about the physiological basis of how we come to measure the signal within functional MRI compared to its background noise. So fMRI depends on BOLD signal. Now, BOLD stands for blood oxygenation level dependent signal. And so the idea is that when cells fire, right, as I said, cells love to use oxygen and glucose. Right? And so when neurons fire, they are subserved by an increase in oxygen. Now, that increase in oxygen is carried to the brain or the neurons themselves via the bloodstream, right? So, and within the blood, we have these red blood cells, which have molecules known as hemoglobin or HB. Right? Hemoglobin, it starts out oxygenated, right? So that's oxygenated hemoglobin or oxyhemoglobin, and after it's exchanged with its oxygen with the neurons themselves, it becomes deoxygenated, right? So it becomes deoxygenated relative to oxygenated hemoglobin. And so the ratio of deoxyhemoglobin to oxyhemoglobin is what gives rise to the blood oxygenated level dependent signal. So again, the physiological basis of the BOLD signal is an indirect measure of neural activity. It's an indirect measure in that when neurons fire, they require oxygen, which is brought to the neurons by hemoglobin in the blood. And so when the blood flow level changes from oxygenated hemoglobin to deoxygenated hemoglobin, that causes an increase in the MRI, in the functional MRI signal. Okay, so the hemodynamic response, right, which is the change of the hemodynamic levels over time, right, which is the measure of the oxygenation level in the blood, that reflects the lag of the BOLD signal, right? So if you're presenting a person with a stimulus, let's say a visual stimulus, it takes a few seconds for the resultant BOLD signal to peak, right? So we're looking at a several second level of temporal resolution here. And so in the MRI, this is what the MRI looks like. and it requires the use of a gigantic magnet, as I've already said, and so it's very important not to get any metal objects close to the magnet, or else you'll get this missile effect where the closer an object gets, the more the bore of the magnet starts to pull at the metallic object. And so this is what not to do. This is what can get everybody fired, and can kill someone. Imagine if there's a person lying in this bed and the table inside the MRI, and if a careless person were to push in this metal chair, it would get sucked in immediately by the MRI, and then this could kill a person, right? So that's a really important point of the training. Part of the training in MRI safety is that you need to remove all metallic objects on you when you get close to the MRI room, scanner room. Now, but when you're using the MRI correctly, you can observe some very good changes, some very clear changes in signal when in specific areas of the brain when a person is lying in the scanner. Right. So this is an example of these voxels, certain voxels that are active a few seconds after a sound is being presented. And so these are tracking when the sound is on and when the sound is off, when the sound is on and off, and so on. And you can see how some of these voxels are more active when sound is being played and less active when sound is being not played, and so on. And so what you could do is a statistical comparison of each and every voxel in the brain when the sound is on versus when it's off, right? And when you do that, you can get a map of the brain where every voxel is the result of a t-test, which is the statistical comparison of these two conditions: music on, sound on, and sound off. And so that's a very important note, is that when we're looking at these brightly colored pictures of brains with areas that are lit up, what we're actually looking at is a map of t-test comparisons, or statistical comparisons. And the areas that are shown as colorful, or shown as active, are simply statistically higher in activity, right? So it's a result of a lot of statistical comparisons. And so another thing to note about this slide is that the strength of the magnet, right? I talked about T-ness envy, where the different groups of MRI researchers are jealous of each other, are jealous of researchers who have a higher strength magnet. And one reason for that is it gives you a better signal-to-noise ratio, right? So for the same kind of study, if you're looking at a scan inside a 3T scanner, it gives you, you can see how these fluctuations are higher. So this means that it gives you a better signal-to-noise ratio at a 3T scanner compared to a 1.5T scanner. But the important point is that the t-tests, these patterns of activations, are simply results of t-tests, right, which are statistical comparisons between different conditions, which are then overlaid on the structural MRI T1 template. Right, so the blobs, right, the colorful blobs come from the functional images themselves, whereas the structural image on which it's overlaid is actually a structural image which is just the same as the T1 image that we've already talked about when we talk about structural imaging. And so from these patterns of activity overlaid on structural templates or structural images, you can also do a three-dimensional visualization. So of course you'll acquire these functional MRI scans at many different slices, right? So it's a three-dimensional volume of different images, which then you can visualize in a three-dimensional view of the brain, and you can also inflate this visualization, right? So when you do that, you're essentially visualizing the brain with its sulci and gyri as an inflated balloon, right? So what you cannot see in this example, for instance, is how there might be some activity that's buried inside a sulcus, right, which is a deep fold within the brain. But when you inflate the whole brain image, you can see better the activity inside the sulci. So these three maps are really of the same data, but they show increasing amounts of data. Now, having talked about how the physiological basis of signal to noise, of both signals, make the signal to noise ratio visualizable in fMRI, let's talk about how we design experiments for fMRI studies. Now, there are a few different major types of fMRI studies. The simplest of which is a block design. And so a block design of a functional MRI study is simply where you get exposure to a number of different stimuli of the same type, followed by different exposures to different levels of stimuli, right? So let's say if you're interested in how the brain responds to dogs versus cats, you might have people look at repeated presentations of dogs, right? So A here is dog and B here is cat, right? So you show dog, 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 dog for 30 seconds, then you have people wait for another 30 seconds, and then you show them cat, 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 cat for another 30 seconds, and so on, right? So you can have these alternating blocks of stimulus presentations. So this is why it's called block design, right? So you're getting blocks of different stimuli. And it's there's some pros and cons of this. There's simple design of a functional MRI study. Now, it's also parametric, right? Because parametric just means you can manipulate the strength of the stimulus at different levels, right? So you can have different kinds of dogs and things that are, if you're interested in brightness, for example, you can show low levels of brightness and then medium levels of brightness and high levels of brightness, and that's a parametric design. And so you can get a good view of the correlates of activity, not just of on and off areas themselves. Now, in contrast, for some of the cons of a block design functional MRI is that you can have low temporal sensitivity, right? So it's pretty much impossible to differentiate in this design between your neural responses to this presentation of the dog versus the next presentation of the dog versus the next presentation, and so on. And another perhaps kind of fundamental problem with the block design fMRI study is that you could get fatigued or habituated to repeated presentations of the same type of stimulus, right? So what if your brain changes in how it responds to the dog, the 10th time it's presented as opposed to the first time, right? So you would not capture the overall strength of the of your comparison in a meaningful way if the nature of your brain response itself changes every time you present as a result of your experiment itself. Now, to use the block design technique in a more nuanced manner, some researchers have actually made use of this ability or a tendency of the brain to habituate to repeated presentations of the same stimulus, and this is known as repetition suppression. And so in a standard functional MRI experiment, you might have a rest trial followed by a first presentation of a stimulus, followed by another rest trial, followed by another presentation of a stimulus, of a different stimulus. But in contrast, you can build in this repetition suppression into the fMRI design, where you can have a first stimulus and then a second repeated presentation of the same stimulus, and then a presentation of a new stimulus, right? So, and the idea is that if a certain brain area can tell the difference between stimulus A and stimulus B, you should see a release from repetition. So this is called repetition suppression. And so this is a more nuanced way to look into how the brain responds to repeated stimuli. So in contrast to block design, event-related design for functional MRI studies is another way in which you can better visualize how the brain responds to certain stimuli. And so in contrast to block design, for event-related design, you present different stimuli in an interleaved manner, in a more spaced out way, right? So instead of having 30 seconds where stimulus A is being presented repeatedly, you present these stimuli in a relatively spaced out over several seconds for each stimulus, but you present them in an interleaved way, right? So here B is interleaved within different presentations of A and so on. And so what you can do then is you can obtain an isolated hemodynamic response function, which is shown here, in response to stimulus A, right? So every time you get a stimulus A, right, you scan immediately afterwards, and you obtain this whole hemodynamic response function. After many presentations of the stimulus A, you can average across all these different presentations and get your average hemodynamic response function. Same for stimulus B, right? And so this gives you a better temporal sensitivity for how the brain responds to stimulus A versus stimulus B. The cons is that you have less data over time, right? Because you're not presenting as many repetitions of the same stimulus over time, because you have to wait a little bit longer between successive presentations of stimuli. But a major pro is that you can have reduced fatigue levels, so you don't have this habituation effect that is associated with rapid repetitions of the same stimulus. And so using this event-related functional MRI technique, you can get functional MRI responses that are related to each event, right? Such as of seeing a face, for instance. And so this is just an example of mapping the visual cortex. We'll talk about vision much more in a later lecture, but you can see how if something is presented in a right hemifield, so right visual space, you get more activity in the left hemisphere and vice versa, right? So just using this event-related fMRI technique, you can see that there's the visual cortex is mapped to visual space. Now, for this next exercise, I'm going to show you some brain images, and I'm going to ask you what you think these people are doing, right? Given these brain images, what do you think the people were doing? What was the stimulus they were given? So this is what we'll call a reverse inference exercise. So, okay, here we have, what's this? This is brain, template brain, with some very clear activity in the superior temporal gyrus. Now, so this is you might think, that if you're a little bit familiar with neuroanatomy by now, you might know that this is where your auditory areas are, right? So this is person was possibly listening to something, right? Now, what about this one? This is a brain with visual activity in the visual areas, so you might expect that the person is seeing something. So what we're doing here is the act of reverse inference, which is the act of inferring the stimulus given the brain activity. In contrast, this is different from forward inference, where you're trying to figure out where in the brain you will see activity given a certain stimulus, right? So forward inference is trying to figure out where the brain activity is given that you've seen a certain stimulus. Reverse inference is trying to figure out the stimulus given brain activity. So these are fundamentally logically different, logically reversed types of trying to understand, trying to draw conclusions from data, right? But if we're starting out as neuroscientists, right, we're trying to, you know what you're giving people, and you're trying to find out where in the brain something is active. you should see activity. But this, what we're doing here, reverse inference, is a little bit more like mind reading, right? So I could see brain activity and I could guess what you were experiencing. And a really dramatic example of reverse inference is comes from this paper, kind of groundbreaking paper by Owen et al. In 2006, where they did scan a patient who was in a vegetative state, right? So they were not moving, they were in an affected conscious state. They were not of full conscious awareness in the ward, but what they were able to do is get this patient to go into functional MRI, and in response to being asked to imagine playing tennis, they actually activated their motor areas, specifically the supplementary motor area. This is very similar to a group of control subjects, and in response to being asked to navigate in the spatially in their own home, they activated the same set of areas as in controls. So based on looking at brain activity of a patient in a persistent vegetative state, Owen et al. Were able to conclude that they were aware, right? So they weren't completely unconscious, that something was getting in there, and that they were able to obey verbal commands inside their brain. So this is a really important use of reverse inference. Let's move on to talk about data analysis and statistical inference in functional MRI studies. Now, we've already shown this slide before, and this is the standard way to do statistical comparisons using fMRI data, right? So you're taking every single voxel of the brain and you're doing these t-tests. You're looking at its fluctuations over time, or you're looking at its activity over time, and you're doing these t-tests to compare activity during stimulus presented versus stimulus not presented. Now, another way to look at this data is to isolate each voxel and look at its time series, right? So these are changes in activity over time. Each of these is a different region of the brain, right? Left and right, middle, and superior temporal gyri. It doesn't really matter for now, but what I'm trying to show you for now is that there are high correlations between these regions, right? So when one region is active, the other is also active. And so this correlation is what's used when we're talking about functional connectivity between different areas, right? So when one region is active, the other is also active. And so this means there's a high correlation between pairs of regions. And so this is one of several ways we can infer that different regions are functionally connected. It's not to say that they're structurally connected, right? It doesn't mean that there's a direct neural connection between one region and the other. It just means that they tend to be co-active at the same time. And this use of different time series is important for classification of patterns of brain activity which that might be active at a certain time. And so the standard kinds of functional MRI analyses are univariate measures, in that you're looking at each voxel independently, right? For each voxel, you're doing a t-test for when something is on versus when it's off. Now, in contrast, you could do a multivariate study where you're considering responses of voxels jointly, one group of voxels at a time. So you're looking at the joint pattern of response between different groups of voxels. So this is known as multivoxel pattern analysis. In popular press, you might have heard of it as brain reading or mind reading. It's also been talked about as pattern classification or prediction and decoding. So this just gives you an intuitive example of how it works, right? So consider so each of these is a brain, right? With each little building here is one voxel within the brain. Now, consider conditions 1 and 2 together, and then consider condition 3. Now, let's look at condition N. Now, do you think condition N is more like condition 1, 2, or 3? Well, most of you probably think that condition N looks more like condition 3, right? But you don't really have to count individually how high each of these how highly active each of these voxels is. You're just appreciating that for condition N, the bright ones and the high ones tend to be on this side, as opposed to on the other side, right? So you can tell just by the joint pattern of highs and lows of activity that condition N is more like condition 3 than it is like conditions 1 and 2. So this gives you an example of how we can do a multivoxel pattern analysis technique, right? So you're looking at the joint probability of activity between different voxels, right? So if you have a little bit of data, right, you can start to see, if you have two voxels, for instance, you can see how when the brain with its two voxels is responding to condition A, you tend to have, lower activity in voxel 1 and more activity in voxel 2. But in contrast, when exposed to condition B, you tend to have more activity in voxel 1 and less activity in voxel 2. Now, with the third condition, it's very hard when there's a little bit of data, when there's not enough data, it's very hard to tell whether the third condition belongs more to condition A or B, but as you have more and more data, as you have more and more voxels, you start to be able to better tell the difference between condition A and condition B. So using this support vector machine, you don't really have to know how to use this or what this really is, other than it's a kind of machine learning based technique that considers the joint probability of different patterns of activity within different voxels, and that can give you a classification for a new condition, whether that belongs to conditions A or B. So this multivoxel pattern analysis of functional MRI data has been used to tell apart, for instance, the difference between brain activity during the visualization of different objects, right? So for instance, you can present a bunch of different objects to the brain, right? Some can be, let's say, bottles and shoes, right? So every time the brain is seeing a bottle, it's showing this pattern of activity. But every time the brain is looking at shoes, it's showing that pattern of activity. And so you can do this kind of discriminant analysis with a large set of voxels, and you can learn, right, over time, given a new scan, right, where it's not any of these training scans now, but you now have a test set, and you can use this test set you can use these training sets to predict the test set with a certain level of accuracy. And so this is done as a kind of really exciting and hot topic of research right now in cognitive neuroscience. You can estimate a model that gives you a receptive field, or gives you a model for each voxel, predicts how likely it is to respond to certain things, and then use that model to make new predictions and to generate a strong prediction of a new object that you're seeing. So using this technique, Kay et al. in 2002 were able to 2008 were able to guess, to predict what a brain saw given its level of patterns of brain activity. And so this is really as close to mind reading as it gets, right? So towards the end of today, I'm going to ask you to watch a video and have a discussion about its implications for how we understand the mind and how we understand how we think and what is possible to do to understand how other people think. For homework, please see the instructions video on Moodle, but I'd like you to do a functional MRI meta-analysis in using both forward and reverse inference. So go to the website. I did a version of it in the other instruction video, which I've already posted on Moodle, but I just want you to think of a term that you'd like to use, a search term, and do a meta-analysis using the Neurosynth website, and then figure out where you're seeing activity in the brain and write a paragraph about your findings. Let's move on to talk about neuropsychology. neuropsychology is the study of patients with neurological and psychiatric disorders. And when you're looking at a patient with a neurological disorder, typically, if you're in a neurology department, you only get an hour with the patient, and 45 minutes of which would be spent taking a history, right? So that means trying to understand the patient as well as you can. And for patients with brain differences, such as neurology patients, it's much more difficult to examine to figure out where exactly they might have brain damage, as opposed to looking at heart or lungs, right? So the heart is either beating or not beating, right, or irregular beating. But with the brain, there are 1,000 different ways it could be going wrong, right? And so your job as a neurologist would be to complete a neurological exam to try to find out the pathophysiology, right, and the anatomical localization of your brain damage or your brain disorder. And that might give you then a diagnosis, and using that diagnosis, you could try to figure out a treatment. So how does a neurological exam work? Usually, when you're going through a neurological exam, you'll be tested for a number of things. The first of which is cranial nerve function. And then after cranial nerve function, you'll also get tested for your motor system and sensory system, and motor system, so coordination and balance and gait, and also for mental status. Now, what do we mean when we're talking about cranial nerves? The cranial nerves are a group of 12 different nerves that are emerging from the bottom of your brain, right? And so they connect your brain towards other parts of the central and peripheral nervous system. So you don't have to memorize all the different nerves right here, but this is just a snapshot of all the different, well, you can't see all of them right here, but the first one, for instance, is olfactory nerve, right? So if you have damage in your olfactory nerve, you can't smell, right? You could also, if you have a damage in your oculomotor nerve, oculomotor means eye movement, right? So you'll show some abnormal patterns of eye movement. So this, for example, is a very clear example of oculomotor nerve damage. If you have damage to your facial nerve, which is cranial nerve 7, you'll have facial cranial nerve damage, which it shows up as a very characteristic kind of abnormality in your face, right? So a kind of facial paralysis. So these are a couple of major signs of cranial nerve damage, right? So if you have somebody like this coming to your neurological ward, it becomes quite clear what the damage might be. But then beyond the cranial nerve exam, you can also do a number of different tests, one of which is a mental status exam. Right? So you might, for instance, test for orientation, right? So for instance, can you tell what year it is, what month it is, what date it is? You can tell ask for registration, right? So do you know where you are, right? You can test for attention and calculation techniques. You can test for recall, right? So some simple memory tests, right? So things like, can you remember 3 objects, hat, apple, pen, and then ask for that again, right? You can test for language and reading and writing, and then a 3-stage command such as, can you do 3 motor movements with your hands, or can you bring this thing over and then put it on the table and then go towards the wall, right? So that's a 3-stage commands. You can also test for construction, right? So the ability to put things together, put words together, and put objects together. Now, a very good example of a person who shows up in the mental as a little bit forgetful in the mental status exam might then be transferred for an MRI, and you might then get something that looks like this, right? So a tumor, for instance, in the frontal lobe can show up in the neurological exam as memory problems. And beyond the level of simple mapping of individual subjects towards different pathophysiologies, such as what Paul Broca did, right? And you're looking at historical cases of like famous neuropsychologists. Paul Broca is kind of the father of of neuropsychology. He was first to identify brain differences that are linked to behavioral differences, right? And so this is at the level of the cortex, and so this leads us to conclude that language might be in the inferior frontal gyrus. Now, what are some problems with this kind of interpretation? One is that you're looking at a sample size of 1, right? So it might be that this person just happens to be odd anyway, right? It might be that he just didn't have language problems anyway. And of course, you also have a lack of premorbid testing. So that means you don't know what patient Tom was like before he showed up to Paul Broca's office. And a third, maybe less obvious problem with the interpretation of this simple one-to-one mapping between symptoms and brain damage is that is something known as diaskesis, which is the loss of function in areas that are distal to the damage. Right? So just because you're looking at this particular region of the brain and that is damaged and the person doesn't have a specific function doesn't mean that this region is responsible for that function. It might be that there's another abnormality somewhere else in the brain that you're not seeing, but that leads to damage in this specific part of the brain, right? So it's kind of hard to conclude just based on a sample size of 1 that just because there's co-occurring difficulty, co-occurring impairments in cortical structure and behavioral symptom, that those two are causally linked. So that's what diaskesis is. And so more modern techniques of neuropsychology are using what's called voxel lesion symptom mapping, or VLSM method. Now, the VLSM method is a more systematic way to relate brain images to behavior. And so this is done using a combination of behavioral assessments, and ideally before and after brain damage, but usually just before, because it's hard to know. When the brain damage might occur in the future, but it's a combination of behavioral assessments and imaging. And so after the researchers have gotten these behavioral assessments and imaging data, what they can do is then draw some lesions and try to identify where exactly the lesions are in each patient's brain, and then register the different brains across different human subjects, and then use that co-registered brain, right, so the average brain with its lesion patterns for analysis and for visualization and then interpretation in relationship to behavior. So this is one nice example of a lesion overlap, right? So each row here is one patient, right? So you can see how there are many different patients, each of whom has different. Some patients have bigger lesions in their brain, some patients have smaller lesions in their brain, but across all these different patients, they share some damage in the frontal lobe, right? They share less damage in the posterior part of the frontal lobe, but all of the patients have damage in the middle frontal gyrus. Right? So that's one example of lesion overlap. Now, what you could do then is color code the brain for the proportion of your patients who have lesion damage in each voxel. Right, so that's a voxel-based mapping between lesions and symptoms. Right, so for every voxel in the brain, you're color coding it by the proportion of your patients who have damage in that voxel. And then you can relate that map to symptoms. And so you could say, for instance, that with certainty that like 100% of your 100 patient sample show who have damage in the inferior frontal gyrus also have problems with generating speech, right? So if you can make that voxel-based lesion symptom mapping claim, then that gives you more confidence in the belief that a certain area of the brain is related to a certain symptom. Just want to end with showing you a video of mind reading in fMRI, and it's from 60 Minutes, the news show. And I would like you guys to discuss a little bit its implications for how we understand the brain and behavior.